Hello, everyone, and welcome to the ReProgram podcast. I'm your host, Dr. George Murphy. Today, we'll be discussing a series of topics very near and dear to my own heart, stem cells and regenerative medicine. Our guest today is Dr. Gustavo Mostoslavsky, my friend and colleague, and together we have worked in these exact fields for more than 2 decades. Dr. Mostoslavsky received his MD from the National University of Tucumán in Argentina and his PhD in cellular and molecular immunology from the Hebrew University in Israel. I hope you enjoyed today's discussion, and remember, knowledge is power. So Gustavo, it's fantastic to have you here today. Thank you for being on the podcast. I suppose we should start with sort of the seminal question, which is specifically how we would define a stem cell.
Well, George, it's great to be here. Thank you for inviting me. I think this is very important because the term stem cell, everyone likes it. It's a hot topic. Everyone likes to talk about stem cells, but reality is, I think that in most cases, people don't really understand what is a stem cell. And this is what we teach. This is what we work with. So let's see if we can get some of these definitions for people to understand it. So a basic definition of a stem cell is actually based on the properties of a stem cell.
Sure.
And these are two main properties. One is that the stem cell can make more of its own, more stem cells, and that in theory, it can be done forever. So that has a very specific technical term. It's called self-renewal, but basically means that a stem cell can make more of a stem cell. And that's one property. And the second property that defines a stem cells is basically the ability of that stem cell under certain conditions, under certain circumstances to differentiate, meaning to change its identity to become another type of cell. And that differentiation changes that it's not anymore a stem cell, but it can become basically, depending on the type of stem cell, any other cell type that we have in our bodies. And that differentiation process is the second property that defines a stem cell.
Thank you. That's super important. So I think as we know, as stem cell biologists, we hear about stem cells all over. They're glamorized in movies. We see things on Instagram. Everything seems to have a stem cell component in it. It's a hot topic and, used as a commercial vehicle to sell products that we would consider to be snake oil. So it's important that we adhere to this sort of classical definition of a stem cell, which, as far as you and I are concerned, as far as our center is concerned, as far as science is concerned, is the most stringent definition. But that definition is often, I would say, bastardized to other things which don't define that stem cell. So perhaps we could go a little further. And could you give us sort of how we feel stem cells would be classified, maybe the two main classes of stem cells?
So this is very important, actually, because there are, in general, we can say that there are two main types of stem cells. Stem cells that a lot of people hear about, and these are what we call adult stem cells, and these are stem cells that you can find actually in different tissues or different organs of a person or animals. But the reason I say that people hear about it because some of those stem cells, which are real and exist, they actually even become part of normal treatments in the clinics. The one that we know a lot about it, and this is the one that has been used the most in clinics, is what we call hematopoietic stem cells, or stem cells that can be isolated, for instance, from the bone marrow. This is one type of very well-known adult stem cell. And it's true that it can be used in hospitals. People do bone marrow transplantation, and the basics for why a bone marrow transplantation works, succeeds, is because of the presence of these adult stem cells in the bone marrow. The second type of these stem cells that we work a lot with are what we call pluripotent stem cells. And now this is very different. And this difference is not actually trivial. And the reason I'm saying this is because a lot of what we have been mentioning on clinics offering, and we're going to talk more about it,
Sure.
I think, it has to do with this concept, with this definition of the difference between what adult type of stem cell is and what a pluripotent stem cell is. Now, pluripotent stem cells, because of, and in the name, it's, the power to make everything. There are very few actual real stem cells that are pluripotent. The gold standard, I would say, is what we call embryonic stem cells. They were very well known many years ago when they were first discovered in 1982 for mouse embryonic stem cells, 1996 for human embryonic stem cells. And these are truly pluripotent stem cells. Now, how do we know how we call them pluripotent? Because In vitro, in a test tube, we can make these cells become almost everything that is in our body. We cannot do this in vivo. We cannot use these cells, that's actually illegal, to use these cells to make an entire organism. But these cells are pluripotent. There was a major discovery in 2006 by a Japanese scientist. His name is Shinya Yamanaka. He got the Nobel Prize for it, where he found a methodology in vitro to make pluripotent stem cells out of not pluripotent stem cells like blood cells or skin cells. And that was a major discovery, because working with embryonic stem cells, you need to actually use an actual embryo. And that has a lot of complications, including ethical, including practical. But the discovery in 2006 by Dr. Yamanaka, how to make these induced pluripotent stem cells, that we work with them all the time here, iPSCs, it really was a game changer in how we approach stem cells, how we can study these pluripotent stem cells, and we can use it for a lot of different things.
Fantastic. So there's so much to unpack in those last few sentences. So to review, I would say that we classify stem cells very simply maybe into two categories, right? So you have adult stem cells, or we would call them tissue-specific stem cells. You brought up one of my favorite stem cells, the blood stem cell, which has the ability to not only make more of itself, but to recapitulate all of the blood cells in our system. And then another complete class of stem cells, maybe we would call them master stem cells. We use the word pluripotent in that it can sort of make any cell or tissue type in the body. And these are sort of stem cells that have been highlighted by the use of embryonic stem cells. Up until the discovery by Yamanaka, embryonic stem cells had sort of moral and ethical implications in their use, and there were governmental implications and things of that nature. With the advent of iPS cells or induced pluripotent stem cells, that opens up all these new sort of therapeutic and sort of biological discovery avenues. So those are absolutely fantastic. To review and maybe talk a little bit more about adult type stem cells, right? Maybe less controversial, but there are controversies that are associated with them, right? So for us, if we adhere to sort of these classical definitions of what a stem cell is, there's a bunch of controversies there. Maybe we'd want to bring up mesenchymal stem cells, which you and I would refer to as mesenchymal stromal cells. Why or why not would you define MSCs, which probably a lot of people have heard about, as not being a stem cell, for example?
This is a great point, George. The term stem cell tourism has become very well known for us in quite a few years now. And this is something happening all the time. People are desperate to find treatments for some devastating diseases, and they will do like anybody else. They will do anything in their power. A father trying to find a cure for a child. And I think that unfortunately can be taken advantage of. And stem cells became a big topic on that regard, because many clinics are offering treatments. And some of these treatments are based on these mesenchymal stromal cells. Mesenchymal stromal cells are cells that have been used and studied for many years, 3 decades at least. And we will define them probably mostly like fibroblasts, because they do look in many regards as just cells that are in different parts of our bodies. I think that, I hope you agree with me, there is no actual evidence in all these 3 decades of work that these cells actually are real stem cells in vivo in the body. It's true that you can force them to become a few different things in vitro, but that doesn't make them stem cells.
Sure.
Now, many groups have done clinical trials using these cells, and the reality is that after all these years, what we know for sure is that these cells cannot become something else. They cannot repair a injured heart or defective muscle, or recover a pancreas, which is the tissue that is affected in diabetes, for instance. But reality is that many companies will advertise they can actually sell you treatments based on these cells to repair tissues and organs. I mean, this is crazy if you ask me.
No, I agree. And so I think it's a super important point. I think for our viewers to review, we've dealt with the concept of the mesenchymal stem cell, which we have now, and I think many people in the field would refer to as not a mesenchymal stem cell, but maybe as a mesenchymal stromal cell. For sure, these cells may have enhanced characteristics, but they do not meet the stringent definition that we defined at the beginning of the show. They don't self-renew in the same way, and they can't make the repertoire of cells in their particular lineage. I think what's really troubling in the general public is the nomenclature of stem cell is used very frequently as far as what you mentioned, as far as treatment options. What we know as actual stem cell biologists is that there are right now lots of hope, but very few actual treatments. And that's problematic. Yes, there are ways in which you can use blood stem cells in the clinic, and there's lots of new and emerging ways in which we can use those cells. There's lots of trials that are on their way, but as of now, these are limited trials, right? So I think one of the things we hear a lot about from Joe Rogan or from sort of other sort of luminaries in the field, or even from athletes, are having these stem cell treatments, using that nomenclature to fix their knees, to fix their backs. And obviously, they would say that there's great sort of results or efficacy that's seen there, right? So I think that's problematic. I think one of the ways I would define if there is clinical efficacy, because sometimes maybe there is that these are definitely not real stem cells, but maybe make a site of inflammation or response where an actual stem cell can then localize to that area and maybe go through some regenerative or reparative mechanism. But for sure, it's not a primary action of that particular cell. I don't know if you have any comments about that.
I may be blunt about this, George,
sure.
but I do get calls and emails constantly from family, from friends, from friends of our family in many countries. And again, it has a lot to do with desperation. So people are desperate to find treatments, and they will go all the way to try to find those treatments. And I think that, to be very blunt, there is no actual cellular treatment right now that will repair a damaged tissue. That is a fact. And I may get in trouble because of this, but that's a reality.
No, I agree.
Now, I think that all the trials, if you read about the results and you can understand the results, in some cases, you can see some effect, like you said, that probably because the cells maybe are secreting some anti-inflammatory agents, that might be true. But you cannot repair a cartilage in a knee because you're injecting cells. That's not happening. And it's very important, because I do believe that in some cases these clinics are using this promise in a very bad way. And people can even sometimes pay a lot of money for something that will not do what they're promised.
And as I think Gustavo noted, we definitely get emails all the time about people that, want to play a role in our research, are willing to be guinea pigs. It's very sad, right? And now we have form emails that we can send out to people. But I think something that's important for the general public to understand is that there's a lot of false claims out there, right? And so people would say, oh, I'm not going to fall for that. I'm educated. I understand these situations. I can go on the internet. But as you noted, people will willingly suspend disbelief if they have a sick child, if they have a sick parent. It's impressive in sort of, how far people will go to try to save one of their loved ones. And that is suspending disbelief and kind of going along with these charlatans. I think the last thing that we can mention as far as stem cell treatments and stem cell tourism is that for us, it used to be all offshore. You would see these people, and still now people would not be doing these things in the United States, but they would go to other countries where, the FDA approval mechanism doesn't work, things of that nature. But I think a troubling trend is that you're seeing that now in the United States. There's lots of clinics in Florida that are doing very similar things. A lot of them are getting in trouble and being exposed, but it's still very problematic. So the last thing I think we would say is for people that are investigating potential stem cell therapies or trying to understand things that might be coming down the pipe that might be actual therapeutic measures, real science, real stem cells used in these therapeutic measures, our agency is something called the ISSCR. I'll put it in the show notes, the International Society of Stem Cell Research. I think viewers can go to that webpage and really understand, again, what a stem cell is, what actually is available, what are the things to avoid out there, and go from there. So, okay, fantastic. I think that's a great overview of one of those classes of stem cells. As we noted, adult stem cells or tissue-specific stem cells. I'd really like to further unpack, though, pluripotent stem cells, maybe the foundational mechanism for us forming our center, really amazing therapeutic potential. I think you're sort of a great historian of the birth and use of induced pluripotent stem cells. So if you would, Gustavo, can you actually take us through the birth and evolution of how the iPS cell came to be? That would be fantastic.
Wow. Okay. That's a great topic. It's very dear to my heart. I started my career with iPSCs when I became a professor here at Boston University. So many scientists were trying to do this, right? To trying to create a pluripotent stem cell out of a differentiated cell or an adult cell, and it was really surprising on one side, but also kind of an amazing, elegant way of experimental setting that Dr. Yamanaka, with his student, found a way just by only forcing expression of only four genes, and that was kind of a surprise. Many scientists thought it was going to take much more than that. But he basically showed in a beautiful way that expressing four genes, an adult cell like a blood cell or a fibroblast from the skin, will literally go back in time and will change completely his identity. And that's why it's called actually reprogramming, nuclear reprogramming. You're reprogram the identity of that cell, and that cell now becomes a pluripotent stem cell that in all regards is almost identical to an embryonic stem cell. This is something incredible because you need to understand first, we adults, we don't have pluripotent stem cells in our bodies. That's a tricky question. We even ask our students when we teach this in class, do we have pluripotent stem cells in our body? And the answer is no, actually. We don't have pluripotent stem cells in our bodies. This is a very, very short window during development in an embryo that these pluripotent stem cells exist. So this discovery is major because now we can make pluripotent stem cells from anyone. Just a little bit of blood or a small piece of skin, and you grow some fibroblasts, you force expression of these four genes, and amazingly, these cells now change the identity to become pluripotent stem cells. Now, this happened in 2006. He did it first in mouse cells. And then, again, a lot of people thought, okay, this is not going to work in human cells. And only a year later, Yamanaka already proved it, that it can be done also in human cells. And it's not surprising he got only in 2012, very few years later, the Nobel Prize for this discovery, because it was really a game changer in the field of stem cells.
Fantastic. Such a great evolution. I remember being back in the lab when that cell paper came out. I'll put the cell paper in the show notes. And, even we were skeptical, right?
Yes.
As when we saw something of this nature. I think now when people go back and look at that paper, probably the cells weren't fully reprogrammed. Again, reprogramming is the process by which you take an adult cell and turn that into a master pluripotent stem cell. So probably in the first paper, it wasn't exactly right, but the technology has been so pervasive. Obviously, the birthplace of the cell is Japan. Syra in Japan now is this master operation of just production of the cells, but also maybe the therapeutic applications, which we'll get to in a second, but really just a groundbreaking discovery. And I think it's the adage that sometimes the simplest ideas end up being the most impactful. Like, why didn't anyone kind of do that before? It's not a very complex experiment, but the idea generation, the ability to sort of pull it off the way Yamanaka did was truly extraordinary. And I think one of the points to reiterate is when iPSCs came to be, that removed all of these moral and ethical dilemmas, because now you didn't need to use embryonic tissue anymore. You didn't need to use a fertilized embryo to make an embryonic stem cell. And depending on how you define life, that would require destruction of the embryo to make an embryonic stem cell. Induced pluripotent stem cells, again, like you said, we have pioneered the use of a tiny bit of peripheral blood to make these master stem cells. Very straightforward, non-invasive, no moral and ethical implications as far as where the cells actually come from. But the amazing thing is you can go from a blood cell or a skin cell to make this master stem cell through this process of reprogramming, and then that cell has the ability to make virtually any cell or tissue type in the body. It's tremendous. I think the other thing that we should mention is once these cells are made from someone in the undifferentiated state, when it has all that potential, we routinely grow these cells in mass forever in that undifferentiated state. So once we sample from someone, make the master stem cell line, we can actually freeze them forever and share them with investigators or people around the world that might want to use a particular line. So one sampling, you can create this master stem cell line. You have the potential to make any cell or tissue type, an unlimited amount of those tissues or cell types. So it's truly amazing. So I think to flex back to the concept of, stem cell tourism, how aberrant that's become. Obviously, when you think about what you just mentioned as far as pluripotent stem cells, the amazing therapeutic potential of these cells. Can you talk a little bit about that?
Well, yeah. This is something that every time I think we present, someone in the audience will ask, okay, so when are we going to be using these cells for therapies? And the answer we like to say all the time is sooner than we think. That we think, because we really are seeing how fast the field has been advanced. And when I say fast, of course, it's in terms of science, right? 20 years is nothing in the field of science. And I know it's a lot of years for patients waiting for therapies, but that's a reality. Science takes time. Science needs to be in a serious way. It needs to be careful. It needs to be with all the controls. And that's the only way that really science can be safe to be used in patients. And what's happening with iPS cells, I think that there was a very big wave in the beginning on the most immediate use, which was a lot of what we do here at the center, which is disease modeling. And this is in part because very quickly we could make iPS cells from a lot of individuals that have particular mutations, for instance. And if these mutations affect particular tissues or organs, then you can make from these iPS cells a tissue type that represent that organ or that tissue. And I can mention a few of them. We can make cells that look like cells of the liver called hepatocytes. You can make cells of the pancreas. You can make cells of the lung. You can make cells of the blood. We can make cells of the gut. And now a lot of people, of course, also doing neurons that are the cells of the brain, or even going farther. And we're going to talk about organoids that have become more fashionable, I guess, in the last 5-6 years. But what we could do very quickly was making these cells out of individuals with mutations, and then use these cells to study the diseases. So this field of disease modeling really was changed by the discovery of iPSs, because before you needed to use other models, and in general those models were not as close to actual human cells like iPSs. But human iPSs are primary cells, and by that, this is an important point I want to mention. We call this primary-like because these are normal cells, and these are not tumor or transformed cells, which is something that people have used for many years to study diseases. Now we're using cells that look actually like the real cells that are in our bodies. And that makes a major impact, a major difference in how you study a disease.
For sure.
So for disease modeling, we have been using them for many years now, and we still do using that. But the question for therapy. So I think that a lot of technologies need to come together to really be able for us to use these cells for therapies. And when I say that, I mean, for instance, bioengineering. The whole field of tissue engineering, who has been in parallel evolving, it still need to get there. And it needs to also, we need to find better ways to collaborate with people in the field of tissue engineering to bring use to our cells. But what I do want to mention, and this is something also you can find in a specific website that you can put in your notes, there has now been more than 100 clinical trials using pluripotent stem cells in general, that includes embryonic stem cells, but around 40, I think, clinical trials using iPSC-derived cells. So it's not zero. A few years ago, we were nowhere even close to that. But things are happening. I think Japan is a leader, like you mentioned. They've been using very, very specific applications. The eye is an obvious one because it's easy to reach, it's easy to follow. So the eye disease has been one of the main targets. But there are a few others as well. And also in America now are becoming more and more used to, I guess, to use the clinical side. I'm going to let you present that.
Yeah, so we basically just pulled the data. As you noted, Japan, the birthplace of the iPSL, has sort of been the leader. If we go to clinicaltrials.gov, which again, I'll put in the show notes, basically you can look at all of the trials that are ongoing. So again, there's this potential is the key word, potential therapeutic use, where this is now being rigorously tested by scientists and for approval from the FDA. There's basically 35 to 45 trials that are currently ongoing, with many more coming up. So basically the numbers, if we look up the numbers on clinicaltrial.gov, there's basically 35 to 45 around that number of clinical trials that are actively using iPS-derived cells. And this is globally. So it's really something that's coming to fruition finally after all of these years. I think no one really knows what the outcome of some of these trials would be. Obviously, there are very specific safety issues that might be involved. but I think the potential therapeutic applications could be transformative. They may transform the way in which we treat patients and understand disease as a whole. I think what you noted is obviously potential therapies. Again, none currently exist. I think that's super important for
Yeah.
the public to understand. And these are things that may come in the future. But right now, not even just right now, but really for maybe the last 10 years or so, as you noted, we've been using these cells, like many investigators, to model diseases in a dish. So this is not the future. This is something that we're doing now that I think one of the biggest hallmarks of the gravity of this and the utility and the fact that it's becoming pervasive in the field is that pharma companies who are always sort of risk averse now have IPS units where they're using IPS cells for drug discovery. We've trained a lot of
Yes.
people that have gone on to fantastic careers in the pharma or biotech industry where they're IPSologists and they're sort of bringing their trade to pharma companies. And so I think it's really started to transform the way we have validated drugs or gone through biological discovery to find, say, novel therapeutics or drugs, right? So the old way that it's been done is there's a series of tests that are, done on a transformed cell line that represents one human. Maybe that's then tested in mice. And then when that's applied to a diverse cohort of people maybe suffering from the same disease, the trials are often fraught with failure because you haven't taken into account the diversity of the patient population you're trying to treat. With iPS cells, we have the ability, we always say a clinical trial in a test tube, you can recapitulate the diversity of the subject population you're trying to treat from a personalized point of view, with the concept being if a drug works in a dish on the cell type that's impacted by the disease, then that will also work in the human the line was created for. So it has this tremendous utility to not only be much more efficacious than conventional drug discovery, but maybe cheaper in the long run and more specific and personalized to the patients we've always wanted to treat. So nothing available coming down the line, but certainly lots of reasons to be optimistic as far as how we might sort of use these cells in the future.
Maybe I want to add to that,
Please.
George. So, like I mentioned before, a lot of technologies are coming together. We cannot not mention, for instance, gene editing, right? So it's not the future anymore. We are doing this routinely now. A lot of laboratories using gene editing to modify, and this is a technique I'm going to clarify. It's a technique that has become, relatively speaking, very easy to do in the lab, where you can literally manipulate the genome. You can actually change the genetic code in a very precise way. That was something that wasn't before possible, right? For many years, we have done gene therapy, the concept of gene therapy, where you introduce an entire gene, but the ability to actually precisely change the genetic code, really the use of CRISPR technology, that's one of methodology for gene editing, it really allows for that. So this is one of those other technologies: tissue engineering, gene editing, cell stem cells. So we are really getting to a time where we're putting together all these different technologies, and it will take years to develop, to be able to merge them. When we say we can make cells of the gut or cells of the lung or cells of the liver, it's not making a liver. It's not making a lung, right? A lung has more than 40 types of cells in an organ. That complexity that takes to make an organ, we are not quite there yet.
I think that's a perfect segue into a discussion about organoids. People have probably heard sort of the concept of an organoid, right? It's not an organ, as you just discussed, but it's an organoid. Can you maybe describe
this is
what an organoid
another
is?
incredible, actually, ability of this stem cells. If you kind of let the stem cell do their job in vitro without doing much, they will find a way to make different cells inside one, organoid. And this was the next level for us in the stem cell field. We were trying for many years to find the perfect protocols, the perfect way to make one type, right? To make one neuron, to make one astrocyte, which is another type of cell in the brain, to make one hepatocyte, to make one pneumocyte of the lung, to make one megakaryocyte in the blood. But then we want more. right? And what happens is, like you said, in many tissues, in many organs, there are many different cell types in that organ. So um, slowly in the field, we have found that if you find the right conditions, the stem cells will make a way to make not one, but different cell types within one organoid. And so in the recent years, more and more we have been using these organoids in different categories. Organoids, for instance, brain organoids, which is an amazing, even think about it, right? It's almost like science fiction, but it's not fiction anymore. We can make these very complex brain organoids now in vitro, where when you analyze them, and they take many months to grow. This is not an experiment that in a week you can analyze. You need 6 months to a year to. Now we've been seeing, even if you let grow these organoids for 2, 3, even 5 years, they do look different. They actually mature more, which is amazing from a biology point of view. But they're very complex organoids. They have 10, 12, 14 different cell types that are normal cell types that you find in a maybe a primitive brain, but you do find all these different type of cells that are normally found in a brain. So I think the term "the sky is the limit" is literally applied here, because we again, this is a relatively still young field, iPSCs, and we can see a lot of really quite an amazing array of discoveries that are coming out of these pluripotent stem cells.
Such an important point. I think as scientists, people always think, we make the right decisions, or we get there immediately the first time and the experiments always work. I think what we always tell the trainees is that, 9 out of 10 times your experiments aren't going to work, and then it's back to the drawing board. I think for us as a center that does regenerative medicine, the interesting thing is that many of us, many of the investigators here, spent more than 10 years, as you noted, figuring out, using human development as a guide, how to make in a dish a blood cell, or how to make a liver cell, or how to make a lung cell. And we figured that out, right? The sort of complex mechanisms that you needed to sort of use from developmental biology, all coming together with all these different growth factors that you add in to make a perfect blood cell or a perfect hepatocyte, something like that. Nothing is perfect, right? The really interesting thing was that's a process we refer to as directed differentiation. We're directing the differentiation of one of these iPS master stem cells into this particular cell type. So we patted ourselves on the back. We thought we'd done a great job. And certainly there are uses. These things have utilities. But then what we realized is that, you know what? That's not the way that nature does it. We always say, people always ask us, are you trying to, be like Mother Nature? We'll never be as good as Mother Nature. Mother Nature will always be best. And we are very crudely trying to recreate these developmental processes. So I think one of the things that we realize, and the field realizes, that cells don't grow in isolation. They grow as complex tissues where you have things that are produced by other cells that impact other cells that demonstratively impact the way something develops over time. And so then it turns out, in the formation of organoids, what's really interesting is we sort of had to back off. So instead of doing this directed sledgehammer approach differentiation of the cells in a dish, if we backed off the growth factors, the pushes that we were giving these cells, and kind of let nature take over, as you really elegantly described, these cells could self-assemble and form structures that were very like the organs and tissues in us, because that's sort of what nature does. So I think that's sort of an incredible thing that we continue every day to learn from nature with organoids. Now we have assembloids where you can actually take different types of tissues and put them together. You can put vascular systems into organoids, and maybe even immune cells, and have different types of systems all sort of operating in the same type of organoid. Like you said, the sky's the limit. I think maybe one of the other things to note is one of your colleagues pioneered the ability to maybe make fully fledged embryos or assembloids that are from embryos in a dish. Maybe you'd like to describe that, speaking of the sky's the limit.
Wow. Yeah, that was another major discovery that happened relatively recently, a few years ago. A colleague and friend of ours, Jose Polo, who was working in Australia, again, kind of serendipitously, he was working with trying to make iPS cells. He noticed the formation of this, he called it blastoids. It was basically the formation of a whole embryo, starting from these pluripotent stem cells, and which is kind of incredible. Again, I need to mention this, right? These are pluripotent stem cells that we know it can make all the tissues and organs, but biologically speaking, we didn't think they would be able to form an entire embryo. And yet, what Jose basically found is that using the right conditions, these cells can actually make an entire embryo. And an entire embryo meaning also including what we call the extra embryonic, which is the placenta that supports the growth of an embryo. And this is, of course, has major implication. Now, ethically, I think that we are still arguing, and the different institutions in different countries are still deciding exactly how to regulate this, right? Because it brings endless possibilities of where you can do in vitro. But let's assure actually that countries are regulating this, right? For a lot of application that we knew before for embryonic stem cells is the same thing. So how many days you can even grow this in vitro, and you need to stop the experiment because it's illegal actually to grow more than certain amount of days. Um, but it does allow now to do things that you couldn't do before, to really look into the actual biology of development very, very early, which was almost impossible to do in other ways. So these cells are still surprising us in many different ways. I do believe that there is a lot of interest on trying to get these cells closer and closer to actually using it, and it's happening, like we mentioned, with the clinical trials. I just hope that people will be patient. It's always difficult, right? Especially when it involves, patients and their desperation to find treatments. But we do need to be patient. Science get there.
Yep.
In history have shown that you can never stop the advance of medical progress. You can't. But it takes time. It takes time and years.
Yeah, and I think what I always think about, and I try to impart to the trainees or the public, is that we are extraordinarily lucky in that we get to be scientists in an era
Mm.
of these transformative methodologies and technologies where we have a plethora of things at our disposal to try to understand biology and science and development, right? So that's really a privilege. And the question I think we've gotten to at this point is, just because we can, should we, right? So I think there's philosophical and ethical debates now because the science has advanced so much where almost everything is possible. We have to take into consideration, should some of these things be done? How should they be regulated? So I think one of the things that I'm always adamant about is we as scientists can't just exist in our sterile enclaves and not get involved in the politics or the processing or the policy making. We need to be advocates for science and the advancements of science, but also be careful as far as how these techniques that can really transform society as a whole will be used and I think accessible to everyone, right? So that's the other thing, is that one of the things we haven't mentioned is how costly. We know from a center point of view how expensive the reagents that are required to perform these basic experiments. Forget about these potentially transformative clinical therapies, extraordinarily expensive. So these are going to be very high-end medicines that are going to have these massive transformative effects, but they're also going to have a massive and maybe inaccessible price tag. So I think that's important for us to also take into consideration.
Yes. Well, but with time we know things get cheaper, things get better, easier to do, more efficient. So again, like everything else, if you think about what was the cost of a bone marrow transplant when it started, what it costs today, and the same thing for many other technologies. So I think that eventually we will get there. And the whole advent of iPSCs, I think what is we will give us is a possibility. That there is a term now that we've been using a lot, which is universal off the shelf.
Sure.
That means by having the cells ready, because we are making them out of iPS cells, that will bring the cost of a lot of the therapies that we're doing today much lower. So
can you explain that a little bit more? So maybe the difference, I'm going to use some technical words, autologous
yes,
versus
okay, that's a great
allogeneic,
point.
right?
Yes, it's a great point. So I'm going to give you a couple of actual real life examples. Okay, there is a relatively new treatment available now for a certain type of cancer called CAR-T. And I know it's technical, but I'm going to go into it. It's going to be easy to understand. This CAR-T is basically take advantage of your own cells that are part of your immune system called T lymphocytes. And what it basically doctors do, they collect these T lymphocytes from your body, from your blood, and they make them better by modifying them like we say, using gene editing outside the body. This is very costly. It takes a few months to do, and it has some complications, but it works. In many cases, it works. And because of that, some type of cancers that were before impossible to treat, now they're actually treatable. So it has really revolutionized the way we can treat some certain forms of cancer, and it's called CAR T. But it is very costly. It is complicated to do, and it uses your own cells. That's what's called autologous.
Right.
And what the field is going towards is, can we do something similar, but it doesn't require now to be the cells extracted, isolated from each patient. It doesn't need the 4 or 5 months of gene editing and gene manipulation outside the body, and with all the complications that happen. And that future way of doing it that we mentioned is this universal off the shelf. Can I now take iPS cells, make these T cells out of an iPS cell, make billions of them, freeze them, and have them ready to go? So that's where we're going. And there are now several companies that are doing it. So many laboratories in the world, including ours, are working on making those cells available. And there will need to be a lot of manipulation, because if I use an iPS cell that comes from somebody to make T cells that are going to somebody else, these cells will need to be modified, right? So they're not rejected. But we're working on it.
Yep.
We now kind of know exactly what we do, and it's getting better and better. It started with 1, then 2, then 3 genetic modifications. Now there are companies doing 16, 20, even 50 genetic modifications in a cell, so they will not be rejected. So the field is going there. The technology is getting closer and closer to that purpose. So again, I think that what you said, the word potential, is really, really what this iPSCs is all about.
So I think we've gone through the journey of what a stem cell is and how we might apply these, the tremendous therapeutic potential being the right word. People need to be very careful as far as the charlatans that are out there and the snake oil salespersons, as far as the fact that these are still therapies that are on their way, but are not ready yet. And people need to be very careful and investigate the backgrounds of things. We talked about master stem cells, what we call pluripotent stem cells, cells that we love, that we use every day, that have not just these therapeutic potential, but the ability to instruct us as far as how nature goes through development, where we can actually, ask and answer basic biological questions, model diseases in a dish. Maybe they're going to revolutionize the way that we do, drug development and validation. And then maybe even develop therapies that might be off the shelf, which might have accessibility to everyone based on the fact they've been modified in such a way using some of these really fancy engineering tools to do that. So I guess my last question to you is, this all already sounds like science fiction. And when we have discussions with people, or when they come to the center to see these things, they're often amazed that these things are happening right now. For you, as a stem cell biologist and regenerative medicine expert, what's the future? So these are things that are actually happening now. Amazingly, there's iPS trials, as we just noted, that are happening, getting ready for FDA approval, that may actually become therapies accessible to everyone. We can actually make organoids, which are kind of like organs, maybe even embryos. What's next? What's the future? If we look ahead 10 or 20 years, where do you see things?
Well, I think that there are clearly certain applications that are coming close to fruition, right? So that's very obvious. Like you said, I will argue, yes, 10, maybe 20 years for those to being approved and used. We mentioned a few of them, maybe for some of the eye diseases, maybe from some muscular diseases. I will like to say that it will take us a little bit longer for what I think the future will be. I have no doubt that these cells will have the capacity to make entire organs. And again, like I mentioned before, when we can put together tissue engineering, bioengineering, gene editing, and the stem cell in the product, I mean, we will be able to make entire organs and tissues, but I think it's going to take us a few more than 10 to 20 years.
So we would call that maybe a bioartificial organ. Can you describe a bioartificial organ and what kind of application that would have in the future?
Well, the bioartificial organ basically will be an organ that we can maybe print or we will be able to put together in vitro by combining all the different cell types that an organ needs, right? You mentioned the vasculature. That's critical, right? An entire liver, if it doesn't have vasculature, it's not a liver. And the same thing for a lung, for a brain. So the moment we will be able to actually recapitulate the entire formation of the organ, including the vasculature, including the immune cells, right? The blood cells, and all the different actual epithelial cells of the organ itself. I'm sure it's going to be done. But people need to be patient, I mean, science is getting there. Science is finding new ways to combine these cells, and a bioartificial organ will be great.
But yeah, in the future, maybe we currently have issues with the number of transplantable organs. That's something that could be obviated by this technology, again, in the future. Anything else you'd want to mention as far as what might be coming down the line, what you see the field doing and
I will
going?
ask people, especially families that are desperate to look for treatment, please educate yourself. Please go to the right sources to read about it. You mentioned a few of them. These are very important sources. The ISSCR has been doing work for many years now to put a very comprehensive list of things you can read about. You can find treatment. So please don't just run and go to the first thing that people offer to you, especially if they're asking for a lot of money for it, because in many of these trials, actually, it doesn't even cost money, right? A clinical trial is people volunteer to participate, and they can participate in these clinical trials. So maybe be suspicious when someone is asking you for a lot of money to try to give you a treatment that sounds too good to be true, basically.
Fantastic. Gustavo, thank you so much for a very enlightening discussion. Thank you for your thoughtful answers, your passion, which is definitely palpable. It's been great being your friend and colleague for the last 20 years, maybe even longer.
Thank you, George, for having me here.
Yes, it's been fantastic. I think in future episodes, we will also discuss the concept of reprogramming, right? From a longevity, from an aging point of view, there's this really interesting nexus between what we do, regenerative medicine, and aging biology, where this process of reprogramming, which in essence is age reversal at the cellular level, is now being thought about as a way to reverse age, maybe in tissues or maybe in full organisms. So I think that's definitely a topic for another podcast. Thank you so much for tuning in today, and please follow our podcast. Remember, knowledge is power. Thank you.